In the concluding class of my series on the development of urogenital system, today I'll be discussing the development of genital ducts. Before we go to today's class, we'll take a quick recap of what we have studied in the previous classes on the development of genital system and gonads. We have studied that sex differentiation in the human embryo is a complex process involving many genes, and Y chromosome is the key to sexual dimorphism. And we also seen the importance of the SRY gene, which is present on the short arm of Y chromosome. The protein product of this gene is called as a testis determining factor, and it's a transcription factor which initiates a cascade of downstream genes that further determines the fate of rudimentary sex organs. So, under the influence of SRY gene and the testis determining factor, the male genital organs develop, and in the absence of SRY gene and testis determining factor, the development of female genital organs is established. We have also seen in detail the development of male and female gonads. To summarize, the development of male or female morphological characteristics are evident by the end of 7th week of development. We have studied that the gonads initially appear as a pair of longitudinal ridges called as genital or gonadal ridges, and these are formed by proliferation of epithelium and condensation of underlying mesenchyme, and germ cells appear by the 6th week. We've also seen about the primordial germ cells. The primordial germ cells originate in the epiblast. They migrate through the primitive streak, and in the 3rd week, the primordial germ cells reside in the endodermal cells of the yolk sac close to the allantois. In the 4th week, the primordial germ cells migrate via the dorsal mesentery of the hindgut, and they reach the primitive gonads in the 5th week. And in the 6th week of development, the primordial germ cells, they invade the genital or gonadal ridges. If the primordial germ cells fail to reach the genital ridges, gonads do not develop. So we can say that the primordial germ cells have got an inductive influence on the development of the gonad, either into the ovary or into the testis. Shortly before and during the arrival of the primordial germ cells, the epithelium of the genital ridge proliferates and epithelial cells penetrate the underlying mesenchyme. Here they form a number of irregularly shaped cords, which are termed as the primitive sex cords. So this denotes the indifferent stage in the development of the gonads. So in this stage, in both male and female embryos, these primitive sex cords, they remain connected to the surface epithelium. So at this stage, it is impossible to differentiate between a male and a female gonad. In embryos with an XX chromosome configuration, the medullary cords of the gonad regress and secondary generation of cortical cords develop, and this forms an ovary. Whereas in embryos with an XY sex chromosome complex, the medullary cords develop into testis cords and secondary cortical cords fail to develop, and this forms a testis. Now we will go to the development of the genital ducts. Coming to the specific learning objectives of this class, first we will discuss about the mesonephric and the paramesonephric ducts. Then we shall see regarding the molecular regulation of genital duct development. Then we'll be studying the genital ducts in males and the genital ducts in females. So we'll be seeing the derivatives and the remnants of these genital ducts both in the males and in the females. And we'll be also be briefly discussing about the common congenital anomalies related to development of uterus. Just like the indifferent stage that we have discussed in the development of the gonads, the development of the genital ducts also have a similar stage. So in the indifferent stage of the genital ducts, both male and the female embryos have two pairs of genital ducts. These are the Mesonephric or the Wolffian ducts and the Paramesonephric or the Mullerian ducts. So in the indifferent stage in the development of the genital ducts, both male and female embryos have two pairs of genital ducts, the Mesonephric or the Wolffian ducts and the Paramesonephric or Mullerian ducts. In the previous class on the development of kidney, it was seen that the mesonephric tubules opened into the mesonephric duct or the Wolffian duct. So this mesonephric duct grows caudally and opens into the cloaca. 2 points to note regarding the mesonephric duct we have already studied in the previous classes. First one, the ureteric bud develops as a small diverticulum from the caudal end of the mesonephric duct just before it opens into the cloaca, and this ureteric bud forms the collecting system of the kidney. And the second point regarding the mesonephric duct that we have studied is that the lower ends of the mesonephric ducts get incorporated into the posterior wall of the urinary bladder to form the trigone of the urinary bladder. So the mesonephric tubules open into the mesonephric duct, which grows caudally and opens into the cloaca. The ureteric bud developed as a small diverticulum from the caudal end of the mesonephric duct gives rise to the collecting system of the kidney, and the lower ends of the mesonephric ducts gets incorporated into the posterior wall of the urinary bladder to form the trigone. The paramesonephric duct, also called as the Mullerian duct, arises as a longitudinal invagination of the epithelium on the anterolateral surface of the urogenital ridge. So the paramesonephric or the Mullerian duct arises as a longitudinal invagination of the epithelium on the anterolateral surface of the urogenital ridge. Cranially, this paramesonephric duct opens into the abdominal cavity with a funnel-like structure. And if you trace it caudally, the paramesonephric duct first runs lateral to the mesonephric duct, then crosses it ventrally to grow caudomedially. So, in the caudal part, the paramesonephric duct runs first lateral to the mesonephric duct, then crosses it ventrally, then grows caudomedially. In the midline, the paramesonephric duct comes in close contact with the duct from the opposite side, and the two ducts fuse to form the uterovaginal canal. So, in the midline, the two ducts come into close contact with each other and eventually fuse to form the uterovaginal canal. The caudal tip of the fused ducts projects into the posterior wall of the urogenital sinus, where it causes a small swelling, which is called as the para-mesonephric or the Mullerian tubercle. So the caudal tip of the fused para-mesonephric ducts projects into the posterior wall of the urogenital sinus to form the para-mesonephric or the Mullerian tubercle. We have already seen that the mesonephric ducts open into the urogenital sinus on either side of the Mullerian tubercle. Coming to the molecular regulation of genital ducts development, we have already stated that SRY is a transcription factor and is a master gene for the testis development. The Mullerian inhibiting substance or MIS produced by the Sertoli cells in the testis causes regression of the para-mesonephric ducts in the males. So the Mullerian inhibiting substance, which is produced by the Sertoli cells of the testis, causes a regression of the para-mesonephric duct, that's the female duct system in the males. And dihydrotestosterone stimulates the development of the external genitalia. So in the males, in the presence of SRY and testis determining factor, there occurs a development of the indifferent gonad into the testis, and further, under the influence of the Mullerian inhibiting substance produced by the Sertoli cells, there is a regression of the para-mesonephric duct system, and under the influence of dihydrotestosterone, there occurs the development of male external genitalia. Regarding the development of genital ducts in the male, the testosterone receptor complexes mediate virilization of the mesonephric ducts, and this virilization of the mesonephric ducts results in the formation of the following structures, namely the vas deferens, seminal vesicles, efferent ductules, and the epididymis. And further on, the dihydrotestosterone receptor complexes modulate differentiation of the male external genitalia. So, in the males, testosterone receptor complexes mediate virilization of the mesonephric ducts to form the vas deferens, seminal vesicles, efferent ductules, and epididymis, and the dihydrotestosterone receptor complexes modulate differentiation of the male external genitalia. Now let us see what happens in female embryos. In the absence of Mullerian inhibiting substance and testosterone, the para-mesonephric ducts continue to develop and gives rise to several structures in the female. Most of the mesonephric tubules and the mesonephric ducts degenerate. Estrogens are also involved in the sexual differentiation. So under the influence of the estrogens, the para-mesonephric or the Mullerian ducts are stimulated to form the uterine tubes, uterus, cervix, and the upper part of the vagina. The estrogens also act on the external genitalia at the indifferent stage. So that under the influence of the estrogen, the para-mesonephric or the Mullerian ducts are stimulated to form the uterine tubes, uterus, cervix, and the upper vagina. Coming back to the further development of the genital ducts in the male, except for the most cranial portion, that is the appendix epididymis, the mesonephric ducts persist and form the main genital ducts in the male. Immediately below the entrance of the efferent ductules, the mesonephric ducts elongate and become highly convoluted, forming the ductus epididymis. During further development of the genital ducts in the male. The region of the mesonephric ducts from the tail of the epididymis to the outbudding of the seminal vesicle obtain a thick muscular coat and forms the ductus deferens. So, part of the mesonephric ducts between the tail of the epididymis and the seminal vesicle obtains a thick muscular coat and forms the ductus deferens. And the region of the mesonephric ducts beyond the seminal vesicle develops into the ejaculatory duct. The part of the mesonephric duct beyond the seminal vesicle forms the ejaculatory duct. What happens to the paramesonephric duct in the males? We have seen that under the influence of the Mullerian inhibiting substance, the paramesonephric ducts remain rudimentary in males and the greater part of the duct disappears. Only the cranial part of the paramesonephric duct persists as the appendix of testis. We have already studied that the prostatic utricle, which is present in the prostatic part of the urethra, develops from the united caudal ends of the paramesonephric ducts, and this prostatic utricle is homologous to the uterus and vagina. To summarize, the paramesonephric duct remain rudimentary in males and most of these ducts disappears under the influence of the Mullerian inhibiting substance. The cranial part persists as the appendix of the testis and the fused caudal ends of the ducts form the prostatic utricle. So you can say that the two remnants of the paramesonephric duct in males are 1. The appendix of testis and 2. The prostatic utricle. Coming to the genital ducts in the female, the paramesonephric ducts develop into the main genital ducts of the female in the absence of Mullerian inhibiting substance. So, in the absence of Mullerian inhibiting substance, the paramesonephric ducts develop into the main genital ducts of the female. The paramesonephric duct consists of 3 parts. A cranial vertical part that opens into the abdominal cavity, a horizontal part that crosses the mesonephric duct, and a caudal vertical part that fuses with its partner from the opposite side. The paramesonephric duct can be divided into 3 parts: a cranial vertical part that opens into the abdominal cavity, a horizontal part that crosses the mesonephric duct, and a caudal vertical part which fuses with its fellow from the opposite side. The first 2 parts of the paramesonephric ducts, that is, you can say the proximal unfused part, develops into the uterine tubes. So the first 2 parts of the paramesonephric duct, that is the proximal unfused part, develop into the uterine tubes, and the caudal parts of the paramesonephric ducts fuse to form the uterovaginal canal. The caudal parts of the 2 paramesonephric ducts fuse to form the uterovaginal canal. The caudal end of the uterovaginal canal comes in close contact with the posterior wall of the pelvic part of urogenital sinus, which later on develops into the urethra. The upper part of the uterovaginal canal forms the uterus, whereas the lower part of the uterovaginal canal forms the upper part of the vagina. So the upper part of the uterovaginal canal develops into the uterus and the lower part forms the upper part of vagina. After the two ducts fuse in the midline, a broad transverse pelvic fold is established. And this fold, which extends from the lateral sides of the fused para-mesonephric ducts towards the walls of the pelvis, forms the broad ligament of the uterus. And you've already seen that the uterine tube lies in its upper border and the ovary lies on the posterior surface of the broad ligament. We have already seen that the caudal ends of the two para-mesonephric ducts fuse in the midline to form the uterovaginal canal, and the upper part of the uterovaginal canal forms the corpus or the body and the cervix of the uterus. These parts are surrounded by a layer of mesenchyme that eventually forms the muscular coat of the uterus, also called as the myometrium, and the peritoneal covering forms the perimetrium. Coming to the remnants of the mesonephric duct in females. The female may retain some remnants of the cranial and caudal excretory tubules, especially in the mesotherium, where they form the epiphoron and paraphoron. The cranial remnants of the excretory tubules form the epiphoron, whereas the caudal or excretory tubules forms the paraphoron. The mesonephric duct usually disappears except for a small cranial portion which is found in the epiphoron, and occasionally a small caudal part of the mesonephric duct may persist in the wall of the uterus or vagina, and later in life this remnant can form a Gartner's cyst. So you can say the remnants of the mesonephric duct in females are the epiphoron, paraphoron, and the Gartner's cyst. There can be several congenital anomalies related to development of uterus. One of this is called as duplication of uterus. This is also termed as uterus didelphys. So the presence of two uteri is called as duplication or uterus didelphys. A much more relatively common congenital anomaly is called as bicornuate uterus. So in bicornuate uterus, the uterus is having two horns, the cervix is separated, and the vagina is single. So the bicornuate uterus, the uterus is in two horns, the cervix is separated, and the vagina is single. This usually results from a non-union of the para-mesonephric ducts in the upper portion of the uterus. Before concluding, we'll see what are the important questions from this class. First one is mesonephric duct and its derivatives. Number 2, para-mesonephric ducts and its derivatives. Number 3, remnants of the mesonephric and the para-mesonephric ducts. That means you have to study the remnants of the mesonephric duct in females and remnants of the para-mesonephric duct in males. And number 4, you have to study in detail the development of the uterus and fallopian tube, including the common congenital anomalies of the uterus. So this finishes our series on the development of the genitourinary system. Thank you.